exactly sure how to cover this topic. So it's a little bit of a mixture of ways to talk about this topic. And I'm hoping that it'll have some good discussion. So if there's something that you really want to talk about this paper, I certainly don't need to follow the path that I've set up, but we were going to start with a video, actually, that it's a long video, but we're only going to show a part of it, but it's by Shai Silverberg, so the person that wrote the paper. And I think he does such a nice job of putting the paper in context, and the whole video that is him giving a talk, but we're going to look at the last part of it. So it's on the NIH website, and you certainly should probably watch it all if you get a chance, because it's a nice presentation. So welcome everyone. Amber, do you want to start?
Okay. That's very different from what we've been reading. Okay, so what can we do about it? Okay. Clearly, we have to improve transparency. There's no question about it. You know, that's the evidence is out there. Everyone knows it. Okay. So how do you increase transparency? Well, one important way is to improve review of manuscripts and grants. Okay, review is key. So when I got involved in this whole business, you know, I said, well, I'm not going to change NIH. It's, you know, even though I'm in NIH, it's very difficult to do. Maybe I can change Nature, Science, Cell, you know, maybe that'll work. Okay. So we organized a workshop, and we invited to this workshop editors, funders, reviewers, investigators for a day and a half of discussions of how we improve transparency. And it was a fascinating meeting. The first day was basically all finger pointing. Everyone blamed the other ones, you know. It's the reviewer's responsibility, it's the editors, it's the investigators, it's the funders, everyone else was to blame. But then something magical happened overnight. I don't know what it was. I don't know what they put in their water. But the next day, everyone agreed that we share responsibility and that we all have to work together. Now, I don't know what happened and how it happened. I suspect that it was something I'll show you in a second. But in any case, this workshop led to this publication within a couple of months. It was prospective in nature with all the invitees, not all, most of the invitees to this workshop. Not everyone signed on. And basically it's got this very long title, but in a nutshell, this long title says, we need more transparency, okay? And it's long because every author wanted at least 2 characters in the title. But this paper ended up having a big impact. And I've often thought about why, and I tend to believe that it's because many times less is more, okay? And here we ask the question, what is the bare minimum that we can all agree on? The bare minimum that needs to be reported. Okay? And that's what we focused on. And that somehow resonated with Nature and Science. Cell wouldn't sign on. But within 8 months, Nature Publishing Group made changes to their review process. They instituted 4 changes which are really important. First of all, they introduced a checklist, quite an extensive one, where you have to say whether you reported on different things and where in the document it's stated. Okay. They lifted the restrictions on the length of the methods, you know, you know. I mean, why would you restrict the methods in the first place? Didn't they read Ramon Cajal's book? Right? They hired biostatisticians to help the reviewers and editors evaluate the statistics. And they provided a platform where you could put the raw data. Okay? Pretty significant change. And in fact, just a few weeks ago, they expanded this to additional areas of research. A year after this, NIH, together with Nature and Science, had a second workshop in which they invited more than 30 journals, and they came up with principles and guidelines for reporting preclinical research. Key to this is transparency. Of course, when you expand it, the more you expand things, then obviously things get diluted because not everyone can agree to everything. So it's not as dramatic as what nature did, for example, or science, but it's a step in the right direction. So here we somehow indirectly influenced how review of manuscripts gets done. But all of you, all of us, are reviewers whenever we go to meetings. We listen to talks, we go to posters, you have posters out here, okay? And how do we decide if they're worthy of looking or remembering or reading up about afterwards? You know, we're reviewing what we're listening to. And it's not good. There's a lot of, lack of transparency in all these things. So we had an idea last year, and we came up with this concept that we should perhaps try to increase transparency at conferences. To this end, we organized a roundtable discussion with conference organizers, and then we published this commentary. Nature called it shake-up conferences, but the idea was simple, okay? A typical figure that you would see in a presentation. You've got these error bars, you have no idea what they are. You don't know what the sample sizes are. You don't know how the experiment was done, and this gets flashed up for 25 milliseconds, and good luck. But what if, you had something like this, where you could see the N, you could see the distribution, you could see the number of experiments, and maybe incorporate emojis, where this is randomized and blinded, and maybe it's a confirmatory study, okay? Maybe the community can come up with emojis that they like, and it's interesting, because in retrospect, we found out that people hate this term emojis. I think the young people get it, but the ones who make the decisions, they don't get it. We should have probably called it icons or something like that. But anyway, this is being tested by FASB in their summer courses, okay, in summer meetings. So it'll be interesting to see where this leads to, But that's up to you, your next generation. Okay, so much for that. What about NIH as far as transparency? Well, NIH changed the review criteria as of January 2016. What they did, they made 4 changes. One is when you write your grant, you're supposed to describe how you're going to do your experiments in a rigorous way to minimize bias, okay? We've talked about this for an hour now, so it's clear what we're talking about, okay? This is what you have to do when you're describing your experiments, okay? That's one thing. The other thing you have to do is talk about what is called now the scientific premise, which is a terrible term, misunderstood. People don't know what we mean by it, and hopefully that's going to change. What it really means is you're supposed to describe the rigor of the supporting data, the key data that you're using in support of your application, okay? You don't want to build a house of cards. I mean, you don't want to waste your time. You don't want to waste taxpayers' money, right? You want to have a solid foundation. Now, that's not always the case, but it's okay. We could at least describe, say, well, there's this interesting result that came up. I don't know if it's actually going to, hold up or true, but I'm going to test that as part of what I'm going to do, okay? So you address the issues in what you're using in support of your data. Just to show you an example, this is a key figure from a grant that was funded by NINDS, and I de-identified everything. So basically what you see here, it says analysis of the inhibitory effect of X on Y in hippocampal slice culture, okay? And you look at this figure and it kind of looks impressive. You've got all these bars and stars and, right, looks kind of cool, okay? The last sentence in the figure says, slices treated with 0.001 and 0.1 millimolar X are missing from the 4-week analysis as they had been lost to contamination, okay? Now, does this sentence disturb you in any way? Does it shake you in any way? Anyone? Yes?
There might have been some low-level contamination.
Yeah. That's probably true, but that wouldn't shake me. Yes? Thank you. I'm shaking, okay? What is the probability that every time they did this experiment, the same 2 concentrations were lost to contamination, okay? Right? That's very good answer, okay? In other words, what are we looking at? We're looking at technical replicates. This was probably done once. And those error bars are just technical replicates. They had probably 3 plates in the same experiment. And those stars are meaningless, okay? Reviewers didn't catch this. Right? So it just shows you how careful one has to be in evaluating your previous work. Okay, the other thing that NIH did was to request that you fill out this form. It's not scorable on authentication of p-biological variables. We talked about problems with cell cultures, antibodies. There was just a paper that came out a couple of weeks ago about probes, how bad they are. So we have problems with resources. One has to come up with a plan how one's going to authenticate them. Finally, biological variables such as sex, incredibly important issue. I don't think it should have been lumped in together with rigor because it's not about rigor, it's about doing proper science. Okay? I can tell you that since this was implemented, I hear from many investigators who say, wow, we're finding differences. Okay? And of course, if you're thinking about the clinic and treating people afterwards, if you develop something that might only be good for females, only good for males, that's a lost opportunity. All right? Okay, almost done. Unconscious bias, which stems from we're not training people properly. Clearly, what we have to do is improve training and through education. NIH is supporting projects to try to develop such material. I don't think it's enough, but that can be changed. Increasing awareness, that's one of the reasons I'm so happy to be here, because every time I speak, hopefully I'm influencing at least 1 or 2 of you. Okay, so that's kind of important. Finally, publication bias and the element of chance. That's a difficult nut to crack. Okay, because it requires changing the culture. Okay, we have to stop focusing on the glitter and focusing more on rigor, and that's difficult to achieve. I believe I have ideas how to do that, but everything takes time. Okay, just to show you where I think we should be, and that's the great Richard Feynman, the physicist, who said if you're doing an experiment, you should report everything you think might make it invalid. Not only what you think is right about it, details that can throw doubt on your interpretation must be given if you know them. You must do the best you can if you know anything at all wrong or possibly wrong to explain it. Good luck doing that in this culture, okay? But that's where we have to strive to go to, okay? So, final take-home message. We're all prone to bias, okay? That's the reality. We're told by great people that are long dead. We're all prone to bias, and because of that, we have to critically assess results in publications. Assess our results and assess publications of others, okay, results of others. Not to take them face value, not to look where they published. It's not important. It's what they contain. Rigorously design, execute, and analyze experiments, taking care of unconscious bias. Plan experiments to disprove your hypothesis, not to prove it right, which unfortunately that's kind of what most science is these days. And last but not least, favor large effects. And I could go on for another hour talking about what these last 3 words mean, and we can't do that, obviously. So I'll just end with one anecdote that you can sleep on tonight. The odds of winning the Mega Million jackpot is 1 in 302 million and change. Yet many people believe they will win the lottery, and some people do. The odds of being struck by lightning in a given year in the United States is 1 in 700,000, okay? Yet many people are terrified of lightning, and some people do get struck by lightning. Yet, we as scientists think that odds of 1 in 20 are highly unlikely to happen by chance. Thank you for your attention. I'll be happy to start a discussion.
Thanks, Amber. I'm gonna, yep. Okay, thanks everybody. That was Shai Silverberg, and there is a longer video. Actually, there's several online that you can see of his, but I did put the link in the chat, so it is worth seeing the whole thing. But I wanted to do a discussion of the paper first, and let me do this. All right. So hopefully everybody got to look at the paper. It is the one that that conference. Hopefully, can you guys see my screen with the paper listed? Okay. So this is the paper that came out from that conference, and I think it's interesting that they now, because Story Landis is the first author, they talk about the Landis requirements. Like there are several papers looking at the effects of this paper on the literature, and it has seemingly made a pretty big impact. That's my impression. And when they went back and did studies, the reporting of the 4 kind of major criteria in the paper, this section here, randomization, blinding, sample size, and data handling, called the 4 Landis criteria, have been much more widely adopted. It's not every, though, paper. When they've looked at it, it's about 50 to 60% of papers now have some reporting of the data, of this information in them. So I think we could, you know, spend hours talking about why some papers don't include it, or why sometimes it's reported as negative. I didn't blind myself to the study, which is kind of interesting. But I thought we could just start by talking about overall sense of how people feel this has impacted their research and other people's research. Anybody have any thoughts, comments? Just feel free to, you know, talk, unmute yourself and talk, or you can write it in the chat, but I got to figure out where the chat is if we do that.
So why do we care?
Yeah.
What, you know, so this is Mike Robinson. I just decided to stop in because I've given this lecture a few times at a couple meetings. And I think the question is, why do we care? Why do we care about rigor and reproducibility? Should we just, you know, just ignore all of it and just move on?
So I think, I mean, they make, I think, good points in the paper and about how it's important to be rigorous and reproducible, so that if someone else needs to replicate your findings, that they're available. But also because preclinical studies are ultimately set up so that we can move those studies into humans, we are able to not waste money, to not harm patients, and to hopefully improve patient care.
So I have about 15 articles that I've pulled on rigor and reproducibility, and many of them are produced. I like your answer, but I'm going to be even more pragmatic. They are published by industry, and they are basically saying that what we are publishing in academia is not reproducible. And they are, you know, in major fields like cardiac and cancer, you know, 60% of what's been published is not reproducible in our hands. And so let's look behind the lines, and we get $41 billion from the federal government to do NIH research. Industry is saying to congressmen and senators, it's great that you fund NIH research, but we can't reproduce any of the work. And so Francis Collins has heard this. Francis Collins, from the director of NIH, he's written an article about this and saying, basically, we want to continue to get funded. We better clean up our act. So I can say something that Shai's not allowed to say. He can't really say what I just said, but I've shared with him that I've adapted his talk to sort of say that, because if we don't all do something, we are in the long run probably not going to be able to continue to get the kind of money we get from the government. And then obviously we look at it right now, and scientists don't, science is not even really respected by the public. And we've got a problem. And if we want to continue, they spend $112 billion, I believe, on research through all of the government agencies. That's a lot of money. And we have to sort of, so that's my start of sort of trying to make it so that we all think about it.
Yeah,
But,
I mean, the paper does go into some detail about challenges in reproducing data. And, they describe the fact that, when you have a study in an animal model and it doesn't recapitulate into humans, you don't know whether the animal model is insufficient or you compounded it by basically overstating your findings. So I do think that, personally, I think these seem rather obvious things to do. I see that somebody wrote that, when we, the complexity of the data that we're creating now makes it somewhat more easier. Well, they're calling it bad data, but I would say it's probably not bad data. It's just data that maybe isn't being vetted without a bias. Is that kind of what you mean by bad data?
well, no, I mean, I've got papers, believe it or not, how much can we rely on published data on potential drug targets? I mean, I've got articles that basically are written by people in industry that are saying that they're trying to reproduce what, the typical Nature science paper where they're, the last two figures try to relate the work to something clinically relevant. And they're saying we can't even reproduce the results. Usually what happens in the science review or the NARAN review, gee, this is a great study, but if you could just do this one experiment. And obviously the implication being, you need to get this result in order for this to be interesting. And so, you basically send your graduate student or your postdoc into the lab and they figure out how to get that result, not necessarily intentionally getting that result, but not subjecting the experiments to quite as much control is my impression of how we're perceived.
So you're saying that in the review process, they're not calling out for the basics to be really firmed up. They're more trying to get it flashier. Is that
Yeah, I mean,
of?
I think sometimes they do that, right? I mean, that's why they were all in the room pointing the fingers at each other. The publishers want the highest profile journals. I mean, I think I don't want to get too much in the weeds, but the bottom line is I think we need to remember that one piece of advice to all of you who are writing grants: don't ever write "I intend to demonstrate." I will show. If what that says is either you're studying something that's so clearly established that you shouldn't get money to do it, or it means that you're not going to rigorously test your hypothesis. Sorry to be controversial here. I'll shut up and just let Brenda go on. I didn't.
No, I think that's true. I think that I try to keep myself from doing it, but it's sort of it's tempting to do it, right? The spin of your science. Yeah, I think that this is like the section here where they talk about, in addition to the measures proposed above, better dissemination of knowledge will be greatly facilitated by addressing publication bias. That's kind of what you're saying, and that very few negative outcomes are published. That's one of the sort of results of publication bias. I think as a junior person, you do have to pick your battles. And since most of the people on this call are more junior, I think that it's a good question how much effort you should put into publishing a study with negative outcomes. So there's more senior people on this call too, but I'd like to hear from the junior people, like if you feel like you're really weighing things, would you potentially not publish a negative study? And what do people think about that?
Can I say something?
Sure.
So I had two thoughts on that. One is that neurology does do an issue, I think, annually of negative results. I'm not sure you can debate the pros and cons of putting them all together. But is a venue for that. But I think it really depends what types of questions you're asking. I think it's important to publish negative results in clinical trials. Like, we had good reason to think this would work and it didn't, and people need to know and understand that it didn't. But in the more basic sciences, if I have a negative result from an experiment, that means I haven't identified the mechanism usually. And I don't think that those can be supplementals or additional figures in the final paper, but each experiment is just one piece of the story. And while it's super frustrating when things don't work, you have unless a negative result answers your question, then I don't know that it's worth publishing.
Interesting. So I think what you're saying is that it may not warrant a full paper, but it needs to be incorporated into a paper.
Oh yeah, like for example, if I'm doing, I can only speak very specifically to my work, right? So I study olfactory processing, and we did a behavior experiment. We did go, no go, and there was no difference between the experimental and the controls. And that was not interpretable. I couldn't take those results and use it to inform my understanding of the circuit. So when we finally publish the paper, there may or may not be a place for that study. And I do think it's important to try and put all of the different pieces of data and include them in your understanding of the final model. But a whole bunch of negative data, unless usually all that negative data doesn't contribute to our understanding of the larger overriding problem in basic science. And so those studies, I think, shouldn't be published until they can be interpreted, which usually requires like studies that had more definitive results. I don't know, I could be wrong.
No, I mean, this is a chance to discuss. Anybody else have anything they want to say?
I mean, I think that in an ideal world, there would be a place for those types of findings, but I think that it's really, really hard. I think it's virtually impossible to like submit a paper based on a primary negative finding. So yeah, it's like, it's easier when you can make it part of a story, and oftentimes it's something that like a reviewer might ask for, well, did you try this? And then it goes into the paper because you're like, yes, I did, and it was negative, but we'll put it in the appendix. But I think that there's just not a great, it's becoming, I think, more in vogue to present and discuss negative results, but there's just really not a great place for them at this point. And I think that they are still perceived as sort of being overall like much lower value than a positive result.
I think that the part of the problem is, anything you do takes time, right? And so you have to balance that time commitment to your career, I guess, at this point, especially when you're junior. I do think, though, that publications still, by their page limits and the review process, probably are still pushing us toward not being as transparent as we should be, right? Because even though, he mentioned that method sections are still, being allowed to be expanded, that's not true in all journals. You still have word count limitations and things like that. You can put methods in supplemental in some journals, but not all. So I think that it's a tough situation to be in as a junior person. I think as a senior person, you have a little bit more leeway to put the time into something that may not move you to the next grant. Mike or Howard, you're on the call. You guys have any thoughts about that?
Can I ask you a question as senior people? Do you find that you go back to your negative data sort of more in retrospect than prospectively? Like if you go back to it and say, well, this was an interesting negative finding and we'll build off of it later. Because I could foresee myself like tucking negative results away to like review at some other point or to try to weave into another story.
Well, Brenda knows my history well, starting back at WashU a long time ago. You know, I feel like my career has been a life of negative results, and it's caused me to approach my science very differently, and I appreciated the way that Shai talked about planning your experiments to disprove your hypothesis, and so I never go in to doing my science without knowing my hypothesis and my alternative hypothesis, and just being very excited by whichever one is the correct one. So I don't see, and I've tried not to have so-called negative results, but just take me where the experiment takes me, and not try to interpret my data or say that, oh no, you know, graph 4 weeks, I contaminated all those things just so that the data fits my hypothesis. I think good science, you know, is designed to appreciate not only your null hypothesis, you know, not only your hypothesis, but to generate your null hypothesis at the same time and to follow the data. I hope that's helpful, but that's been my career because I have yet to prove any of my hypotheses. My PhD took way longer than I should because I was wrong.
But you got that PhD.
But I did get that PhD, and I feel better because I worry, you know, we're talking about rigor and reproducibility. I often worry about the graduate student who's up against a deadline. It's been 6 years, and how much of our data is not reproducible because of, you know, seeing what you want to see in the data to get out of the lab, to get that big paper. And I think that plays a big role. I'm working with an animal right now that's a great model of subcortical band heterotopia. And the graduate student who worked on it before me, having a reduced number of parvalbumin cells in the normotopic cortex perfectly explained her data, but so did a failure of development of perineuronal nets. And I can't reproduce her decrease in parvalbumin, but I'm finding a decrease in the number of parvalbumin cells with perineuronal nets. But at that point, Brenda hadn't told us the importance of perineuronal nets yet. So, you know, at that point, her findings fit prevailing hypotheses. And I do think that that when you don't set it up so that you can come out either way, that it sometimes leads you to the wrong result. So I don't know if I'm being cynical or helpful, but that's been my 30+ year experience as a pseudoscientist in some ways.
That was very eloquent, I thought. Very nice. Yeah, I think that there are some really practical things that come out of reading this paper that, you know, they kind of break down all the different spots in your research that you can go astray, right? And not only you, but exactly as Howard said, you know, when you start to run a lab of your own, you have to be the person that's critically looking at the data being produced by your postdocs, by your graduate students. And if anything I've learned is a really bad graduate student is somebody that makes their data fit what you want to see. That's way, way worse than having somebody that comes to you with like, well, it doesn't look like it worked. Great. At least you know that they are bringing you all the pieces of data. You know, somebody that you have that you're overseeing that doesn't bring you all the data can really, really ruin you. So I think it's important to try to instill this into the people working for you. So you should have it and they should have it. And that I think will make for a much more productive career, maybe not the first 3 months, but 3 years or some down the road. So if you start to look at this, you know, and you divide it up into these different topics of randomization, blinding, sample size estimate, and data handling, I think that we've all, or I shouldn't say we all, I have been guilty of different aspects of this. And I think we all probably on occasion are like, oh, I didn't calculate the sample size correctly, right? You know, you get the result and then you're like, do I do another 10 animals or is this really a negative result? So I think they're all really pretty helpful, just basic ideas. Does anybody have anything else that they wanted to talk about with the paper? I was going to go over the nature guidelines, which I think are really a little bit even more crisp about where you can go wrong and what you should be doing. But before we get off the paper, does anybody else have anything that they want to really talk about?
Hi, it's Art Riddle. This is the first one of these I think I've been in, but nice to see you. But I was going to say, I thought the paper was a great summary of things, and I think that awareness of this is certainly, even in my research career, changed the way things happen in the labs I've worked in and the way I do things since the time, you know, when I was a lab tech. And I think make the data a lot more reproducible and trustworthy overall. You know, one thing I think that is a challenge, in addition to just publishing negative data, is publishing data that challenges or is different than sort of existing dogma or paradigms. And that can also be a really challenging, in my experience, in the review process. I think negative data, and I think when I first started in my graduate work, proved, my PIs and my hypothesis wrong in the first 2 months. It was a great opportunity because it turned out that you could then figure out what the alternative mechanisms were. But some of that work was really hard to publish because it challenged sort of the existing thought process. And so, that's something I think about sometimes is, at this stage in my career, do I want to be asking questions that, or I do try to think about it in ways that, framing it in ways that don't do that, because that can be more challenging to get published in my experience. But I didn't sort of hear that side of things, and that there can be certain challenges in the review process in the paper. But.
Yeah, I mean, I think that you're getting to the heart of the matter. Like you want to do good science and you want to make sure you're testing a hypothesis, but that sometimes can lead you down a path that you meet more challenges with, right? And so you long term, I think you're doing the right thing, but you may not get that grant because someone has a strong bias that what you're proposing can't be true. Or there's no way that's possible. And that's sort of the problem. I mean, that is in a nutshell. Mike wants to talk.
I had a classic example of that. Several years ago, we pulled down a glutamate transporter from brain. This glutamate transporter is supposed to be on fine astrocytic processes. All we got was mitochondrial proteins that co-amino precipitated with it. And I had people tell me, very smart people, one of whom was clearly on the study section, tell me that mitochondria couldn't possibly be in fine astrocytic processes. And we proved it, and they now agree. They've published high-profile papers in the same field, but you can persist as long as the science is there. You can battle through that stuff, even if you're junior. I do have one piece of advice for writing your grants, because to me, that's the next most important thing you're going to be doing in addition to publishing. You need to make sure that it's easy for the reviewers to see that you've addressed this. Remember that when you're writing grants, reviewers are probably not printing out your grants. They're reading them online. They can miss stuff. And so you want to make sure that it's easy for people to see, a section, rigor and reproducibility or something, so that it's there and can be found. Because if the worst thing that can happen is that a reviewer misses it, and then all the other reviewers miss it, and then you're basically going down this path of, gee, we can't even discuss it. It has to come back to address this kind of stuff.
Here, let me, well, a failed scientist, I've seemed to have become a professional reviewer for NIH. I guess that's my lot in life. I would plead with you, use these keywords and make it easy for me to cut and paste. I really love when I can just cut and paste over onto my score sheet. If I don't have to think about it, you're going to get a better score from me than if I have to spend time thinking. If I have to think, I'm going to find all the holes in your grant. And so the last thing you want me to do at 11 o'clock at night is to spend time really digging into your grant. So I
Completely agree with that advice. Thank you, Howard.
You're welcome.
It's all about the reviewers when you're writing a grant. It's not about you. It's all about making the reviewer, make it easy for the reviewer. I've chaired study sections. I've been on, I think I've been on over 40 study sections now in my 30 some years of this career. And it's all about the reviewer. Make it easy, short paragraphs, white space, and make it easy for them to see that you've got the information, even if it's in bullets.
So
let me just be, Brenda, sorry, let me give an endorsement for Grammarly. I don't get no money from them, but reading Strunk and White before you start to write your grant is very helpful. And then using something like Grammarly that's going to go through your sentences and say, this one's too complicated, is very helpful.
Oh, interesting.
yeah. Can you address the fact that they added this to the, this rigor and reproducibility and gave you no more space essentially in your grant? And I have struggled with helping, like I was just helping our R25 candidates submit theirs, and those are very short. And I sort of felt like the rigor and reproducibility was in every single section of the grant, right? I mean, it should be there. So when you pull it out and you bullet it, what does that exactly mean to you guys?
Well, I'll share with you with the grant, my most recent grant that was funded, it was all on the last page of the research strategy. And I basically just referred them to that. All the statistical stuff was there, all of the blinding that we would do, all the way through data reporting. And in fact, actually, the statistics was complicated enough that the statistician and I wrote the paragraph together, and the statistician, who Mary Putt, was part of the grant. So
you basically, you put the whole grant together, and then at the end, so once they've kind of been able to kind of understand the grant, then you describe to them what was rigor and reproducible and where you were meeting all these criteria. That's interesting. Howard, what do you think?
Well, I'm sitting here with an R21 application open and trying to get it from 7 to 6 pages. So my rigor and reproducibility for this grant is right before my specific aim one, just getting it out there, saying, here it is. I know I need to do this. I'm thinking about it. And I come back to it every once in a while, like any good grant, you pick those things that you weave and repeat that you want to make sure the reviewer hasn't missed. So in my pitfalls, I come back to rigor and reproducibility every once in a while, just to show the reviewer I'm thinking about it.
I see.
And so much of the grant is just making the reviewer aware that you have spent a lot of time thinking about it, that they don't have to spend time thinking about it because you've already thought about all your pitfalls. You've thought where you might not be rigorous or why you can't be blinded. So there is one section here that there is no way to be blinded. Either the mouse is going to seize in response to temperature elevations or it's not. So, I'm talking about how we're going to have to mark those differently so the person doing cell counts is at least blinded, and that we're going to separate the person doing the experiment for this person doing cell count. And that's in the experimental design of that experiment. So I think you need to weave it all the way through. But I've been doing rigor and reproducibility with sex as a biological variable right up front to say, I'm addressing these. And again, so the reviewer could just put a check mark. Yes, rigor and reproducibility. Yes, sex as a biological variable. And not spend a lot of time thinking about it.
That's very helpful, because I do think that, it's a fundamental part of, oops, sorry, I'm going to mute somebody. I think it's a fundamental part of the grant. And so it's tricky to kind of pull it out without taking up too much space. So I think those are both really good advice.
And I think in the grant that I had it at the end of the section, I also said, this will be addressed at the end. And the reason I did it, I would have normally done it the way Howard did it, but because of the particular grant, they had to see the experiments in order to understand the statistical analyses. There were some, yeah.
Nice. So I have up, I believe, on the screen now the Nature Reporting Life Sciences Research document, which I mean, I've never published in Nature, so what do I know? But I think it's a really helpful document for any paper. So it goes through all the things that the paper goes through, but it goes through it in a lot more detail on the specifics of what you should kind of think about when you are designing the experiments. And it gives you some details, like, you know, how you should, for instance, this section on the reporting statistics, it gives you, you know, you should make sure your distribution on your data is, you know, not skewed. It's, you know, it's got a normal distribution. So you can find it online. So I'm not going to go through it in a lot of detail, but I think it builds on the paper and gives you more like concrete, like do this, do that. It also is interesting, not, you know, as neurologists, a lot of us don't do a lot of cell line work, but it gives you some ideas about, I didn't really see this. Oh, bye, Howard. Thanks.
Bye, Howard.
It gives you some ideas about, like, I didn't realize this, but there's a lot of cell lines out there that are like notoriously not thought highly of, right. Which I think this is much more in the cancer field and other fields, but that was kind of interesting to me that, you know, people know that certain cell lines are not good or stable, things like that. So anyway, I would recommend that you maybe look over this one. And then one final thing I wanted to do is, this is a paper from somebody I've always respected a lot in the field of epilepsy, which is what I work on, Paul Buckmaster. And long ago, long before, well, long before I moved to Stanford for sure, but long before this whole idea of rigor and reproducibility, his papers I always have felt were probably some of the most rigorous, carefully designed science that exists in the field of epilepsy research. And I just want to go through his methods section briefly and sort of point out that, you know, he hits every point that they talk about. So he tells you exactly how many animals in total were used for the entire group of studies, like how many started with, you know, how many he ended up with. He gives you their ages. He gives you how they did the seizure monitoring. They were blinded to the results. So you had one group, like, just like Howard said, you know, it's hard sometimes if you're doing animal studies to blind the same researcher to the phenotype and then to do the analysis. So, and I think that's challenging for some of you guys if you're in a small lab and you don't have enough personnel to really blind everyone to the process. I think that's when you have to really reach out and find somebody to collaborate with, perhaps, or, you know, because I do think it's important that graduate student will probably be biased if they know that animal is, you know, they saw that animal have a lot of seizures. So I think that, you know, he hits all those points. He says that blinded to the animal, the anatomical results. So he had more than one person working on it. He's amazing histologist. Every single, from the tip to the back of a hippocampus is being sectioned and measured. It's a ton of work, but he's always been really methodical about his histology. And then when you look at what they recommend for antibody characterization, you know, each antibody has a paragraph. Like, how often do you see that in a paper where somebody goes through the entire details of every antibody used? He talks about where they've reported it previously, how they've studied it, and whether or not it has specificity. And it ends up taking like a whole page of the paper, which just doesn't happen. But it's that kind of detail that makes you totally trust what he says. So anyway, I just think that there are really key parts that when I read his paper years ago, I always thought, wow, this is really good science. He talks about always trying to figure out whether the data is distributed normally. And if it's not have a normal distribution, he changes his statistical approach. And so you know in detail whether or not there's some difference in the distribution of his data. So, I don't know. I bring that up mostly because I think that this detail puts him like a step above the average scientist in my mind. And I've always been really impressed. So you can bring yourselves to that level, right? You can be the Paul Buckmaster of somebody's life if you do it to this kind of extent. But we're running out of time, but I wanted to give more time to people to talk about anything that came up when they were reading the paper, or anything else that we touched on, but they want to discuss.
Hi, this is Danielle Barber. I'll
Hi.
just chime in for a second. So first of all, thank you so much for sharing that. I will definitely write his name down and look those up, because it's so useful to have an example as, you know, kind of a gold standard or something to kind of aspire to in a practical way. Something that I have recently been talking a lot with my mentor about is litter effects. And how to think, I mean, especially true in developmental work, but also true even in adult, and I'm talking about mice specifically, but could be true for rats or other animal research. Even in adult animals, there's evidence that animals that originated from the same litter are more similar to each other than animals that came from different litters, even across an inbred strain like C57 Black 6. And so we've been thinking a lot about how to design experiments where we're taking into consideration litters in terms of N. And what is an N? Is an N really multiple animals within the same litter or across different litters? It definitely complicates experimental design. So I'm kind of curious if anybody else here is also doing mouse research, especially with young pups, and has been thinking about these litter effects and what kind of approach other people are.
Really good question.
So
Good
Danielle,
question.
can I refer you to Mary Putt? Mary Putt is working with several investigators who have exactly the same issue.
Yeah,
she's available to you through the IDRC, and just copy me on the email. So
thank you, Mike. Yeah, Amelia Amelia is my PI, and
yeah.
Mike knows, has talked with Mary about this, and we actually also within a setting, the behavioral interest group meetings at CHOP/Penn, we've been talking about this specifically. We've been talking about it a lot with Gordon Bart. We're like starting a bigger conversation about this, because I think it's really actually important, and something that again, in the same vein of transparency, often is not reported when people report their work. They don't say how many litters their pups came from.
Yeah, there's actually something, Elizabeth, randomizing on the same litter, different cages after weaning. Use litter cage as a blocking factor. Yeah, exactly. Well, did you see what Elizabeth just wrote? Elizabeth, you can talk, can't you?
I saw that. I don't know if I.
Oh, it's direct to me. Oh my gosh. Elizabeth, are you on the call? Are you?
Yeah, sorry. I'm just trying not to scare my mice.
Oh, okay. All right. Elizabeth says, I'll talk for her then, that what she's doing, I guess, is randomizing animals from the same litter to different cages after weaning or delivery, and then using litter and or cage as a blocking factor in the statistical analysis. So that's one way to get at the fact that you have a biological variable and you think it might be important. So I think your question is terrific. Yeah,
it's tricky because I'm like running into the same thing too. But again, if Elizabeth may be dealing with, she may be able to do it in a different way. But if you're dealing with mice that are younger than wean age, then they still need to be nursing, and so trying to randomize immediately after birth is a little tricky. But actually, in the paper that you sent out, they there was a paper, they kind of in passing refer to like litter effects, and they refer to a paper that I started looking at after reading the paper for today that I found really helpful. So Danielle, it's reference 35 on the paper that Brenda sent out for today's discussion. But yes, I think that that's something that's coming up for me too.
Yeah, I haven't read reference 35 yet, but I've read a couple of other like papers that are trying to deal with these litter effects. And my takeaway from having looked at this pretty carefully is that there's no gold standard approach. There's no one right way to do it. You just, I think in the end, you just have to report, be transparent about exactly what you did, and then whoever is trying to reproduce your work or can take that into account when they're thinking about how to digest that work. I don't know.
Yeah, I think that is a good approach, to be very clear on how you did it. So at least any potential bias could be figured out by the reviewer or the reader. Benjamin K. wrote something, which I don't know what it means, but the sandwich estimator, it sounds like your lunch. I don't know what that is, but anyway, that's sort of a post hoc way to do the analysis. Is that right, Benjamin?
I mean, yeah, so we do this like in neuroimaging data. If we have like households with multiple siblings who are in the research, obviously we like can't randomize that, be like, sorry, you have to go live with someone else. So we just hope there's enough random variability in our population that we can use a mixed effects model where the household would be a random effect to get around that.
And the sandwich part of that, what does that mean?
The sandwich estimator, it's like a computational shortcut, but it does the same thing.
Okay, cool. I was thinking maybe it's because you're sandwiched with someone else, but no, that's not what it means.
That would be cool though.
Okay. Yeah, these are all good questions. All right, we've used up our time, but I thank everyone for participating. We'll have another undecided topic, because I haven't gotten to it, for February. So if somebody has any burning interest, they can shoot me an email. I'll probably try to find something in the next, I don't know, week or two as a topic for our next webinar. But anything else before we go? Okay. Yeah. All right. Thanks, everyone. Bye.
Thank you.
Thanks, Amber.
Sure.